Hello, everyone. Welcome back to my SCENIC video tutorials. In my previous video, we did order analysis. In this video, we start to explore and interpret the results. So let's load the SCENIC package. So as I mentioned before, if you see the files home, you can see in the home directory, we have a folder named inter. This is the folder to save all the intermediate data. We can have a look at this folder. You can see all the RDS data in this inter folder. They are named as 1.1 to 1.6. Those RDS files are the analysis when we did a co-expression network analysis to filter the genes, correlation analysis, and run GNE3. Also 1.5 and 1.6 are run SCENIC step 1 analysis. Then from 2.1 to 2.6 are run SCENIC 2 step 2 analysis. So from 3.1 to 3.5 are the dataset from run SCENIC 3 analysis. In two optional step analysis, 4.1 to 4.4 are the RDS dataset from the analysis of binarized network activity. The 50 PC 30 perplexity RD files was generated from step 3 analysis. Then other t-SNE RDS files were generated by the option step 2 analysis when we cluster the cells based on the regular activity. So the data in the inter files can be loaded into R using the load inter function together with the SCENIC option object. So if we run load inter SCENIC options, you can see we can see all the files in the inter folder. We can use both names to load the individual files. For example, if we want to load the AU cell regular AUC, it is a file from here AUC regular AUC. In the inter folder, the file's name is 3.4 regular AUC RDS. If we run the load int function, the object is SCENIC option object. Then, together with the individual RDS name, we can load this RDS into R. So we can load the AU cell regular AUC files as AU cell regular AUC. When we run the analysis, we also save the result in the output folder. We can open the output folder to have a look at the result. So you can see here, the results were labeled as each step. You can see we have results from step 2, step 3, and step 4. You can also see two folders here for step 3 and step 4. So let's have a look at the result from each step. You can see the step 2, there is a TSV file called the motif enrichment, and also there is a preview HTML for the motif enrichment analysis. We can open the TSV file. You can see the details for the motif enrichment analysis from this demonstration dataset. In the first row is the motif database. We use the 500 BP and 10K BP database. Second row is the gene set for transcription factors that were identified in our analysis. We use the co-expression method top 5% target. So the HTML files has the same information as the TSV file. So let's open the preview file. You can see the possible binding DNA sequence for the identified transcription factors. We have the motif database and also the high confidence and the low confidence transcription factors. So in step 2, it also generated a TSV file named as regular target information. We can open it. You can see all the target genes for each transcription factor, DLX5, IRF1. So we have 6 transcription factors here in this dataset. We will look at the target genes in RStudio as well. You can also see 3 files for step 3 analysis. So the first one is the regular activity heat map. We can make it bigger. So you can see the heat map for regular activity for each transcription factor, including the extended regular activity. So the second file for step 3 analysis is the HTML files for the regular activity on TCNI cell types. Let's open it. You can see there are 4 images for each transcription factor. We can use DLX5 as an example to look at the images. The first one is the threshold for DLX5. Second one is the cells in blue that passed the threshold bigger than 0.08 for DLX5. The third one is the gene set activity in those cells for DLX5. And the last one is the DLX5 expression from the matrix data in those cells. So you realize the TSNI cell clusters are different from the cell clusters that we set for default TSNI assay. So we can have a look at the third files. So that's the TSNI cell clusters after step 3 analysis. So you can see this is the TSNI cell cluster with 50 pieces, 30 perplexity. So this is the TSNI map that was generated in step 3 analysis. Later, when we perform step 4 cell clustering, we set the default TSNI as 5 PC and 15 perplexity. If you don't perform the optional step analysis in step 4, then your TSNI cell cluster based on the regular activity will be this one, 50 pieces, 30 perplexity as the default TSNI map. You can also see a folder for step 3 analysis. So we already see the HTML file. We see 4 images for each transcription factor. In this file, you can find the individual images for each transcription factor. We already explained it, so we can go back to see step 4 analysis. Step 4 will be very similar as step 3 analysis, but we just binarize the network activity. So step 4 will generate more heat map. You can see we have the heat map for all transcription factors. We also have the heat map for correlation, non-correlation, and 1%. So here is the HTML for each transcription factors. After we binarize the network activity, you can see the threshold for the DLX5 cells past the threshold and the regional activity for DLX file in those cells. And also we have the t-SNE map for the binary regional activity. You can see the cell cluster look different as the step 3 analysis. Okay, we look at all the result in the output folder, and also we look at the RDS files in the inter folder. So now we can generate other images and tables to interpret the data in RStudio. So the SCENIC analysis can identify cell states and their regulators. So first we can project the transcription factor on the t-SNE map because the cells were clustered based on the regional activities. So we already know the EU cell regional AUC dataset. Then we can have a look at the default t-SNE map. We can find the default t-SNE map by run t-SNE file name for the SCENIC option object. So let's run. You can see the default t-SNE map is 5PC 15 perplexity RDS dataset. So let's set the figures for plotting in 2 rows and 3 columns. Then we can first plot the default t-SNE cell clusters. So next we can project the transcription factor onto the t-SNE map. So first we need to know the default t-SNE RDS. We need the t-SNE information to project the transcription factor activity. So let's load the default t-SNE dataset. Then we can use the t-SNE information to show the cell clusters and also show the expression for each transcription factor using the expression matrix data. So here we are going to show the regional activity for transcription factor IRF1, SOX9, SOX10, DLX5, and we are not going to show the extended regional activities. So let's run to show the expression for those transcription factors. We can zoom in. You can see the cells were clustered based on the regional activity, and we have the default t-SNE map, 5PC 15 perplexity. So here we show the expression of 5 transcription factors in different cell clusters. You can see IRF1 has expression in microglia, SOX9 has expression in astrocyte, and SOX10 has expression in oligodendrocyte. DLX5 and STAC6 has expression in neurons. So if you want to save the expression images as a PDF file, you can export, save as a PDF. Here you can change all the parameters. So I just show you how to save it as a PDF file. I'm not going to save it. So now we can switch off the settings for figures. Then it doesn't affect the following analysis. Another important function of SCENIC analysis is to identify the cell states. So we can use the density plot to detect the most likely stable states for the cells. So to make the density plot, we need to load the 2 packages, current smooth and Arcana brewer. Now we can make the density plot to see the stable state for the cells. We can zoom in. According to the online tutorial, the high density in the t-SNE map detect most likely stable cell state. So next we can look at the regular target in RStudio. So we already looked at the output files from step 2 to see the target information. Because the regular target information file contains thousands of target genes, you may just interested in some important transcription factors in your study. So we can subset the target genes for interested transcription factor. Then we can export it as a HTML file. So let's first note the regular target information RDS. Then we are going to use transcription factor stat6 as an example to extract the target information for stat6 transcription factor. We will generate a table for stat6 target information. You can see we generated a new data frame. We named it the table subset. We can connect the dataset and have a look at the target information. So you can see we have 66 rows here. So that means we have 66 target genes for stat6. You can see the first column is the transcription factor stat6. The second column are the target genes for stat6. We also have other information, for example, high confidence annotation is true. We have the motif database and the co-expression weight for stat6 and the target gene. So now we can export it to HTML file for stat6. Let's run. And we can zoom in. You can see the possible binding sequence for stat6, the motif information and the target genes. And the other information we just showed it in the table. If you don't want to make a table, you can also just extract the target gene names for each transcription factor. So let's load the regular RDS dataset. Then we can extract the target genes. We are going to use the DLX5 and the IRF as an example to extract the target gene names for both transcription factors. Let's run. You can see down here we have 25 target genes for DLX5 transcription factor. And also we have 59 target genes for the transcription factor IRF1. So for the SCENIC analysis, only resonance with 10 genes or more than 10 genes are scored with AU cell. So let's note the AU cell resonance RDS dataset. Then we can have a look at how many transcription factors were scored with the AU cell. If we run, you can see in this dataset, we only have the regular activity for 6 transcription factors. Actually, there are more transcription factors in this dataset, but only TAF, IRF1, STAC6, SOX9, SOX10, and DLX5 has 10 or more than 10 target genes. After the target genes, we can have a look at the regular motifs. So we already looked at the motif enrichment files from step 2 analysis. You can see from the output folder, we have a TSV file for the motif enrichment analysis, and also we have a HTML file to preview the motif enrichment result. So if we open the preview motif enrichment file again, you can see in this data set we only have 6 transcription factors. Because this is a small demonstration dataset, sometimes in your big dataset, you may have lots of transcription factors that have regular activity, but you are not interested in all the transcription factors. You may just focus on some interested transcription factors for your data analysis. So we can just extract the motif information for an interested transcription factor. So the motif information was saved in the file motif enrichment self-motif W genes. So we can load the motif information. Then we can use SOX9 as an example to extract the motif information just for SOX9. Then we can view the motif information for SOX9. So let's zoom in. You can see here is the motif information for top 5% targets. Here are the possible binding DNA sequence. We have the 500 BP and 10K BP database under the high confidence transcription factors. So I already show you how to look at the cell state, target gene information, and the motif information in RStudio. Now I can show you how to combine SCENIC analysis with other analysis tools. The first one, we can combine SCENIC analysis with complex heat map to show the average regular activity by each cell. Then we can identify the regulators for the known cell types and the cell clusters. First, we need to load the complex heat map package. Then we can load the AU cell regular AUC dataset again. So we are going to make a heat map plot, not include the extended data. Now we can get the regular activity for each cell type. So you can see here is the regular activity for each cell type. We can click the dataset, have a look. You can see we have 6 transcription factors. The extended regular activities for each transcription factors were not included in the dataset, and we have 5 cell types. Here are the regular activities for each transcription factor in different cell types. So you can see the regular activity range from very low level to the highest level 0.73. So we can scale the data in order to make a better heat map. So let's scale the data. Now we can make a heat map for the 6 transcription factors in each cell type. And we are going to use the scaled data, and the legend name will be regular activity. So let's make a heat map plot. We can zoom in to see the heat map plot. You can see the red color show high regular activity for the transcription factor in different cell types. You can see IRF1 STAT6 have high regular activity in microglia. SOX10 has high regular activity in oligodendrocyte. SOX9 has high regular activity in astrocyte. Interneurons are DLX5 and TAF also has higher regular activity in pyramidal C1 neurons than interneurons. So the heat map doesn't show the exact number of regular activity, so we can generate a table to show the number for the relative activity for each transcription factor in different cell types. So we already see the top transcription factor by layer color in the different cell types from the heat map plot. So now we can generate a table for the top transcription factors in each cell type. We can get the data from the scaled regular activities, then name the column names, and show the relative activity bigger than 0. Now we can view the top regulators in a table. So let's zoom in. You can see the name of transcription factors and their regular activity in each cell type and also the relative activity. This is scaled data. Because in the analysis we run the optional step to bialize the network activity, so we can also generate a heat map for the bialized data. So first we can set a minimum percentage. For example here, 40% to only plot the transcription factor that show regular activity at least in 40% cells. So let's set the minimum percentage. Now we can load the AUC cell binary dataset. We also need to get the cell type information and then the regular activity in each cell. So now we can fill the transcription factors with the minimum percentage and also bigger than 0. Let's run. So now we can make the heat map to show the regular activity for each transcription factor using the bialized data. So let's run to make the heat map. We can zoom in. You can see now the red color shows the percentage of cells in each cell type with the regular activity for the transcription factor. Again, you can see microglia, IRF1 and STAC6 are active. DRX5 are active in interneurons and pyramidal C1 in neurons. SOX10 is active in oligodendrocyte and SOX9 is active in astrocyte. Like the above analysis, we can also make a table to show the top regulators and the relative activity for the top transcription factor in each cell type. So let's generate the table. And we can zoom in. You can see now we have the transcription factor, the cell type, relative activity in percentage. So finally, in this demonstration video, I can show you how to calculate the regular specific score to identify the cell type specific regulators. So they are the transcription factors. So let's note the AU cell regular AUC data again. So now we can calculate the regular specific score. Then we can plot a regular specific score to make the plot later. Now we can generate the plot. We can zoom in again. You can see the DLX5 has high regular specific score in interneurons. SOX10 has high score in oligodendrocyte. So STAC6 and IF1 has high score in microglia. For pyramidal neurons are TAF, and SOX9 has high score in astrocyte. So here we show all the transcription factors in each cell type. If you are interested in one cell type, we can also plot the regular specific score for one cell type Here we are going to plot the interneurons. Let's run the plot. We can zoom in. You can see DLX5 has a high score in interneurons. The rest are very low. Okay, let's order demonstration in this video to explore and look at the result. This video is quite long. I hope you have time to watch it and like the video. I'm going to stop from here for the demonstration. Please subscribe to my channel if you haven't to do so. Hope to see you in my other video tutorials.